Okay, welcome to the first part of our genome variant analysis section. That's the GVA part 1. You'll want to go to Canvas to download two things. Let me go ahead and share my screen. You're going to want to get the genome variant analysis part 1 PowerPoint. So go over here. So on Canvas, you'll see in the variant analysis section, there's the PowerPoint, GVA part 1, but then there's also this tarball, the GVA_data. And you'll want to download that because that's the data that we're going to work on starting today. Okay. Okay, so this is sort of loosely based on a course that was originally developed by the Texas Advanced Computing Cluster. They have a genomics computational biology group there. Doesn't bear a whole lot of resemblance to that original course, but it was really nice that they shared some of the scaffolding for that. Most of what I'm going to talk about is based on Illumina data. The exercises that we go through are going to be based on Illumina data because that is the most common platform still used today, because the price per base pair is so low. We're going to do whole genome resequencing and mapping, partly because those are the most common things that you're going to do. Now, some of the labs at UTA in particular are really involved in genome sequencing projects. My lab, for example, when you don't work in model organisms, you do end up running into genome assembly more often. But in the big picture, doing the genome for an organism is kind of a one-off project. And so I have the asterisk there because for those labs that really do this, you realize you're never truly done with a genome. But once you have a genome, most of the subsequent projects are, for example, RNA-seq or any of those are going to involve resequencing and then mapping that to the genome that you've already assembled. So the kinds of things that we're going to go over in class are more aligned with more common tasks. In subsequent courses, you know, in the genomics course, you'll spend a little bit more time on some of the other aspects that might go on related to dealing with big genome projects. Today, we're really going to focus on quality control of the read data, and then in the next couple lectures, we'll identify and then look at variants. Okay. So what are we talking about when we talk about mapping Illumina data? Illumina data comes in two flavors, either single or paired end reads. When we talk about a read, we're just talking about really a small sequence that in a mapping project is something that we want to match to a unique region in the genome. Now, most people have switched to paired end. It's been a long time since I read a paper that did single end sequencing. The main value of paired end sequencing is that it's much easier to map them to unique regions in the genome. We've sort of recognized over the last couple decades that genomes are full of repetitive content, and if you can't map a read uniquely to a spot in the genome, then a lot of times you just have to throw it away. So the value of having paired ends is that you can match read 1 to a place in the genome. And then even if that single read doesn't map to a unique spot, then you have a lot more information because read 2 is also trying to map to a unique spot, and you know roughly based on the preparation of doing the sequencing how much space should be between those, and that gives you more information about the unique location that those two reads might have come from. But the idea between paired end and single end mapping is essentially the same. You're just taking your read and trying to find where did that come from in the genome. Okay. The most common read lengths today, whether you're talking about paired end or single end, is 100 to 150 base pairs. Okay. So when you see some notation like I want the arrow, I want the laser. There we go. When you see notation like 2x150, that is the 2 is for paired end and the 150 is for how long each of the reads is. So you're really getting 300 base pairs out of that fragment. Okay. It's just 150 base pairs on both ends. And then there's some stretch, usually 300 or 400 base pairs in the middle that you're not sequencing. Okay. Sometimes you also see the notation PE150. People are, the literature is moving away from this kind of confusing notation. Well, I don't know, if you grew up with it, I guess, like I did, it's not that confusing, but paired end 150 or PE 150 is also a common way that you'll see that specified. If it's single end, it would be SE 150. If you're talking about a sequencing project, you can break it up into sort of 4 phases where you sort of identifying the biological question. All of these are important. Once you have that question, designing and conducting the experiment. So what kind of sequencing are you going to do? That leads into, how are you going to prepare the NGS's next generation sequencing? The libraries, we're going to talk about that in a few minutes. And then the sequencing, what platform are you going to use? And then number 4, the analysis. Typically, number 4 takes the longest. It is the sort of, in some ways, the most complicated, interpreting what comes out of this. And so that's what we're going to spend, well, plus this is a bioinformatics class. So we're going to spend most of our time talking about, or I'm going to spend most of my time talking about the last step. But that said, I do need to talk about some of the design and prep stages, just so you have an idea of what is the data that we're dealing with. I think that helps understand what's going on in the analysis part. So basic Illumina workflow, you extract your DNA from your organism, fragment that. There are multiple ways to fragment enzymes, sonication, nebulization. Basically, you're just shattering the genome into lots of small fragments. Then you're going to size select, right? Because I talked about how it, especially the most common is paired end 150 base pair reads. Your fragments, you want to be somewhere in that 250 to 700 base pair region. And the smaller the range is, really the better it is for this issue of knowing where does that thing map uniquely. So if you can get a tight window on size selection where everything is, in this case it would be 150, that's 300 base pairs plus 300 to 400. So you're talking about 600 to 700 base pair fragments. Now, if you can make a window that tight, right, now you've got a lot of information. But if your size selection, this window ranges anywhere from 0 to 400, now read 1 and read 2 could be there's more variation, and so there's less information in where that might map uniquely. Okay, so ideally when you do size selection, you're looking for tight windows. Once you've got your fragments selected, then you go into the chemistry of ligating adapters. Those adapters are what's going, so that's the sort of pink and purple dots here in the cartoon. The flow cell is sort of a lawn of oligonucleotides that are complementary to the adapters. And so when you put your ligated DNA fragments onto the flow cell, they're going to anneal with the oligos that are sort of on that lawn. And then they do this bridge amplification. There's a YouTube video here that will walk you through it. I'm not going to spend a lot of time. I think you'll probably see this information. If you haven't seen it in courses already, you'll see this probably many times. The most important issues as far as we're concerned, well, one is the size of the fragments, right? The idea that these are actually relatively small in the big picture of a whole genome. And then the way the PCR is done sometimes allows these things to grow into each other, and so you get these are called clusters. And the way the data is actually collected is you do this amplification on the slide, and it grows clusters of the same sequence near each other. Okay, and then what's happening is something called sequencing by synthesis. Okay, and it's really a camera taking pictures of, right, these clusters. I've got some more slides to show how that works. This picture is blown up version. The high C flow cells are, this is roughly the size of a sort of a microscope slide with billions of these little oligomers all over it. And there have been advances in that technology. We'll talk about the different throughput of the Illumina machines in a second. So how does sequencing by synthesis work? Right, so here, for example, we've blown up and focused in on one single oligomer, the flow cell. These lines are the adapter and the oligo that was tethered to the flow cell. So it's latched here, and we want to sequence this sample. So what you're doing is basically throwing in ACGT that are all have fluorescent molecules attached to them. When the DNA polymerase attaches or finds the complement there, so as it synthesizes the complementary strand of DNA, and then you shoot this thing with a laser, it fluoresces the color of that nucleotide. And you just repeat this over and over again. And at each stage here, you take a picture. So here I'm showing how two different sites on the flow cell would be interpreted. So I've circled them. These are the same two clusters on the flow cell. The first one fluoresced green at stage 1. So did both of them fluoresced green, so they're both T there. But then this cluster fluoresced blue, so it's a G. Okay, and you can sort of follow along. That top dot gets a T, G, C, T, and so on. The bottom one has a long run of T's. It keeps fluorescing green. okay, and then it gets a G, and it goes back to T, okay? So that's, I mean, the chemistry and the actual machine itself, how you get high resolution images of that and in real time, that's all complicated, but in principle, it's a pretty simple operation. This just to give you an idea, the Illumina technological innovations have been expanding rapidly. The example I was showing is 4-channel chemistry, which is on 2 of their machines. The NovaSeq, which is the highest capacity one, actually uses a 2-channel, so only 2 colors chemistry. And then the iSeq, which is a lower throughput, uses a single color. I've given some links here. If you're really interested in how the technology works, you can watch videos on that. It's not super important for our analysis. A lot of times as a bioinformatician, you're getting the data that you're getting off the machine is already translated by the software that's on the machine from the images into the base pairs, okay? And it's also going to give you a quality score. So how confident is it that it called the right base pair? And so you can kind of see up here, I was talking about how here these clusters can grow into each other. And there's a lot that goes into, you know, how much of your sample do you put on there to try to get these clusters spaced far enough apart that they can get big enough to give you good signal, but not grow into each other. And you can see it's a little bit random here, where those clusters grew, and that's based on where these things originally landed and annealed on the surface of the flow cell. Well, one of the advances that has made the throughput higher is that they make these micro wells so that the clusters are separated from each other better, and you can sort of more predictably fit a given number of clusters on a flow cell. You still do get, we call them chimeric clusters, where here you've got 2 that have kind of either grown into each other, or the same well on a plate got more than one molecule ligated or annealed into it. In which case, right, that's going to lead to a low quality signal, because at any given position, if it's got a different base pair, you're going to be getting 2 signals from the same cluster. And then, like I said, downstream of that, what you're going to get as a bioinformatician is a base pair call and then a quality score, which correspond to how confident are we that that was actually the base pair at that position, which is corresponding to how clear is the signal from that flash of fluorescence. Just to give you an idea what kind of technologies are out there and what their capacities are. These are the most common instruments. Well, I shouldn't say that. These are all of the Illumina instruments. The ones that are have squares in red are the ones that we have here at UTA. So the North Texas Genome Center has a NovaSeq. We may also have some high-seqs. I don't remember. There are a couple MySeqs on campus, and then the Genome Center. There's one in the Life Science Core facility and one in the Genome Center, and then there's also an iSeq in the Genome Center. This scale here, where you're talking about so maximum output, how many base pairs do you get off of a run? You can go the iSeq, right? These are relatively low capacity, so 1.2 gigabase pairs to 15. NextSeq sort of bridges a gap. This goes up to 120, but these all sort of fit on your lab bench. Then the production, so Genome Center scale, goes anywhere from that NextSeq, which bridges the gap, to things like the high-seq and then the nova-seq, which is insane. So you can get 20 billion reads, which amounts to about 6 trillion base pairs. Part of the trade-off, just a last note here, is that things like the MySeq that are not giving you very many runs, the MySeq in particular can give you 300 base pair single reads. Okay, so you can go up to 600 base pairs of actual sequence on a given fragment, right? So this is 2x300, right? Remember that's paired end 300. You've got 300 base pair reads on both ends of your fragment. That's as good as the Illumina technology can do. And it's really that they are constrained. This technology is constrained by this whole fundamental way that it works, where you're the sequencing by synthesis needing to grow these fragments. Okay, once they start to get too long, they flop over, and you just, at least so far, can't read them longer. It's also partly the way they shoot it with a laser over and over again, that those enzymes degrade. So haven't had an increase over 150 base pair reads on the production scale instruments in quite a while by genome sequencing standards, like maybe 5 years at least. And then same here, the MySeq, those reads have kind of maxed out at 300 as well. So it'd be interesting to see in the future if these can get go longer or not. How much sequencing are you getting? Just to give you ideas, right? So those are the numbers from that NovaSeq, which is the highest capacity production instrument that Illumina has. You get, if the human genome is 3 billion base pairs, it'd be kind of a waste to do a human genome on one run of that, right? You don't need 2,000-fold, 2,000x coverage of a genome. So there's a question on these really high production, and why do you want to go on a high production? Well, the cost per base pair is significantly lower, like 10 or 100 times lower per base that you sequence on the NovaSeq or the high-seq. So really cheap to get lots of sequence. But if you don't want to waste all of that, how do you put multiple samples on there? And that gets us to the barcoding. And so essentially what you do is, in addition to the adapter that you ligate, you also, on that adapter, put some indexing or barcoding primers. And so what that does is add some sequence that is going to be, or it adds a short string of DNA that is going to be sequenced on the NovaSeq, for example. So when you get your sequencing, right, this is the primer where, for example, if we go back here, this is where the DNA polymerase is going to start, okay? And so if it starts, actually, I should probably go back and revise these a little bit, but in any case, what the index does is, so example, you read through here and it gets either this index or this index, and it depends on which kit you're using. That's why I'm sort of hesitating here. But the idea is that you will have an index that after you sequence it, you can computationally, right? This is the bioinformatics part, demultiplex. So when you add the primers so that you can put multiple individuals, that's called multiplexing. And so you demultiplex by recognizing your index or your barcode, and then that barcode is what tells you this read belonged to sample number 1 or number 2 or number 20. Okay. That whole standard library preparation is fine if you've got clonal samples, right? You've got lots of sequences from the same individual, for example. So you know that every base is going to be 0 or 100%. Meaning when you map it to the genome, every base pair position should be all of your reads from your sample. If the genome position is a T, for example, all of your reads, if it matches the genome, should be T. Or if there's a mutation there, they should all be G, for example. They'll all be this, all your reads should be the same. If it's a diploid organism, obviously you might have 2 variants, right? Because if it's heterozygote, half the reads may come from the T variant, half may come from the G variant, okay? But there are limited possibilities, so that standard library prep works well for that. Pooled populations. So for example, if I want to do a whole bunch of beetles at the same time, because I'm interested really in allele frequency, that standard library prep is fine for that. So rather than do the barcoding, I could just say take 50 beetles, grind them all up together. And why would I do that? Because my beetles are really small. There's not a whole lot of DNA there. And a lot of times when you're working on small samples and you don't have a lot of DNA, you may want to pool that rather than do a separate preparation to index each one of them individually. So doing that is fine. The standard Illumina rate is about 1%. So if you have allele frequencies that are higher than 1% variance, then the standard prep works fine. I'll talk about a little bit more about that in a second, where library prep is not sufficient. Okay, and then mapping, when you're mapping to a good reference genome. So what do I mean by good? It's closely related to the samples that you're using, and it has long contigs or scaffolds. Okay, the more chopped up and small the contigs and scaffolds are, right? So that means there are more breaks in the assembled genome. Then that makes it harder to map the reads, because the reads might span a break or an unassembled part, and then you don't know where the reads belong, and you just kind of have to throw them away. If you have a good reference genome, so you know, the best ones are like the scaffolds are chromosome length DNA, so you know very long stretches. That makes for easy mapping. It's often lacking if you're interested in rare variants. So for humans, for example, if you're interested in rare variant detection, you have to take some alternative library prep methods, because you know, 10% variation might not, if you're pooling DNA from multiple individuals, you won't see things that are less than 10% frequent. Now, again, that's not true if you have DNA from an individual, right? Or if it's a clonal sample, right, all came from the same individual. Because, right, when it lines up, this is just to give you an idea, two variations here. The top line in this cartoon is the reference genome. And now we have a bunch of reads that map to this location that in the reference is a T, but all of the reads from our sample were G. And then you compare that over here where the reference is a C, most of our reads are also C, but then we have a T right there. Well, there's a good chance that's an error, right? Because it only happens once. If all of these came from the same individual, we should get roughly 50-50 C's and T's. Now, we don't have very many reads there, and that's an argument for needing greater read depth, right? If we had sequenced that position at 20-fold, right? So we would say this is 1, 2, 3, 4, 5, 6. This is 6x coverage of that site. This one only has 5x coverage. If we had 20x coverage on both of these sites, then what would the frequency of T be? It becomes, you know, if that stays the only one, that becomes more and more likely that it's just an error in the sequencing read. Okay. Now, if you double right, if we go up to 20x coverage and now we start to pick up 3, 4, 5 T's, now you start to say, okay, actually, that's probably a heterozygous site. If you have pooled DNA from multiple individuals, it becomes very hard to identify those kind of errors, right? Because it could be that only one individual in your pool of 50 had the T mutation. And so you can't tell the difference between an error and just a low frequency variant in the population. So that's what we mean by the error rate in the sequencing technology sets limits on variant detection when you have pooled samples, when there are multiple individuals there. A lot of times that standard library prep might not be good for non-model organisms because we don't have good reference assemblies to map them to. I talked a little bit about that. I'm going to go into a little bit more detail about solving some of the problems here. Most of that issue is with short reads, is because of the short read sequencing, right? When we can only go out, the whole fragment is 700 base pairs long, for example. It really makes it hard to map things uniquely. It makes it hard to assemble genomes. Just a lot of problems that arise, and a lot of bioinformatics, frankly, has grown up around having to deal with short read sequencing. So I'll talk about some ways that have developed to avoid those problems. And then the last thing, if you have a genome or a part of a genome that has really high repeat content, the reads, even the paired end reads, may have a hard time finding unique locations in the genome. And the longer the repeats are, the more difficult that is, again, because of the shortness of the reads. So how do you reduce errors, right? This is the first one, okay? Since the majority of errors are randomly distributed along the reads, if we have reads that are from the same original molecule of DNA, we should be able to distinguish errors from variants. And this is one of the strategies, especially in human biology or human genetics, where we are really interested in those low frequency reads. So what do? Instead of taking a 700 base pair fragment and having 150 base pairs on both ends, we make fragments small so that read 1 overlaps with read 2. And so now we've sequenced the same molecule of DNA twice. And so anything that's unique between read 1 and read 2 must be an error, and everything that is the same would be a true variant, okay? Something that we'll see in our exercise later is that this is a pretty typical quality score profile for Illumina reads. So this is average quality score on the y-axis and then the base pair position. In this strategy, right, what you can see is toward the end of the read, the quality really drifts off, and that's well, not partly. That is because of the way the Illumina chemistry works. The enzymes just degrade, and you get less and less clear signal. So in this overlapping read preparation, you all of these low quality things on the end are going to be eliminated because they're going to be unique to either read 1 or read 2. Okay, and it's only the ones in that region that show up in both reads that you're going to say those are the actual variant. And it's not just, remember, this is not just there is a difference there. It's that it has the same difference. So, for example, the green arrow here in the same position, it means read 1 said there was a G there and read 2 said it was a G there, even though the reference is an A at that position. Okay, whereas the red arrows are saying, okay, on read 1 it called it an A, but on read 2 it called it a G. And then you would say, no, that's probably an error, because you can't get a heterozygote on one individual molecule. Okay. Okay, the second one, how do you improve the length? So this is another strategy. The MySeq talked about that. Mate pair libraries. So this is where you can essentially create a way to get reads of known distance apart that are higher, they're farther away than the fragmenting method that I talked about before. Okay, in this case, what you do is you have a really big insert from your DNA that's represented by the black chunk there, and then you ligate adapters onto the end. Okay, and then circularize the DNA, and then now you know that when you end up reading, so here it's got the Illumina adapters, the purple and blue stuff. Now you know when you read off of that these two black pieces are, in some cases, right, if this is 25 kb, you know that black read and that black read are really separated by a known distance, and that could be a really long distance. So just one way to use the Illumina short read sequencing to get location information or distance information beyond what you could do with the standard library prep. And then the last one, high-C or chromatin conformation capture libraries. I'm gonna say a little bit more about those because these are pretty hot right now. A lot of people have shifted to high-C because it gives you similar kinds of long distance information that this does. It's just that the idea behind this workflow is different. So what you're doing here is cross-linking it, usually with formaldehyde. I can never remember which one. But then you chop up your DNA after those things are linked together. Then you tag the ends, ligate them together, and then pull them down. So with a biotinylated molecule, just drawing blanks today. And so now you know everything that you pull down was in proximity to each other in whatever environment you did your cross-linking. And why I'm vague there is that you can do this in vitro or an in vivo approaches where you're getting genome organization or nuclear organization. The idea, though, is that these things must have been close together, and so when you do those reads, you get, and remember, we're collecting billions and billions of these sort of, you could call them proximity indicators, and that goes into a graph like this, where this is the contact map is showing the number of times that, right, this purple and orange showed up in the same Illumina fragment. Okay, and so on the diagonal here is putting fragments that show up a very large number of times. The sort of the darkness of the red is an indicator of the frequency that things occur. And so the bioinformatics pipeline here is basically saying, can I, what's the, in terms of like the dynamic programming, what's the minimum path to put this whole genome together that maximizes the number of contacts across those fragments? And this is great because what you can see, the blue boxes here, this is for a recent, this is actually the Tribolium confusum genome that we started to look at in BLAST, and we may look at farther down the road. One of the cool things that this shows is right where the centromeres are on these things. Okay. 6 is kind of weird, right? Actually, there are multiple weird ones. This is chromosome X and chromosome 2. We kind of knew there was a fusion of these two things. But anyway, don't want to get lost. So this is called a contact map. This is the basic structure that you are looking for to get out of doing sequencing with a high C. Really good at giving you information about genome organization, or the sort of cooler thing that is maybe not quite so straightforward is that if you're getting this information about how things are contacting each other in the nucleus, that might be telling you something about chromatin organization, which tells you something about gene regulation, at least in principle. Last two are technological innovations to try to get you past the short read problems. One is a technology most commonly referred to as PacBio. It's based on single molecule real-time sequencing. You can get, I think they're actually, they're passing this now. But anyway, you're talking about tens of KB, not hundreds of base pairs. So this is 15,000 base pairs. And there are two strategies for this. One is continuous read. So you'll see CLR, PacBio sequencing, continuous long read. And then there's also an interrupted sequencing where they turn the laser off to try to, because again, the laser is degrading the polymerase along the way. If you leave the laser on to get a continuous read, your read is shorter than if you turn the laser on intermittently. So you read 100 base pairs, turn it off, read another 100 base pairs, turn it off. That's where they can get out to 80 kb or more on a single molecule. You're still knowing all of these reads that you're getting are linked together physically because you're reading literally right along that molecule. The main disadvantages, why doesn't everybody use this then, is that it has a lot lower throughput than Illumina. The last time I looked, they were around 80 gigabases in a run, right? Compare that to 60,000 or 6,000 gigabases per run. This ends up being way more expensive per base pair. So the main reasons you would employ this are those, things I was talking about where standard prep is not good. If you want to do genome assembly, great. If you want to analyze structural variation, great. Genome assembly is moving in this direction again, mostly because of how cheap Illumina sequencing is. You can do multiple sequencing, multiple genomes in a single run, novel genomes. And then the other reason why it has some issues is that it has a high error rate. So the error rate can be, more than 10%. So 1 in every 10 base pairs is a mistake. Okay. I star this that they're random errors because if you get multiple reads, Illumina reads are somewhat biased. I showed you they sort of get lower quality toward the end of the read. The PacBio doesn't have that issue as much. Oh, the other thing is Illumina has trouble sometimes with multiple runs of a single base pair. So if you have a run of A's or a run of T's, for example, sometimes it makes a mistake. Like it will incorporate two A's and you don't know, like the software doesn't know, should it call a single A or two A's? And so when you have a run of 5, and some of the sequences will say it's only 4, some will say it's 5. So you get those kind of errors. Those are biased errors that this PacBio sequencing doesn't have. So you need less coverage in order to correct those errors or to see where those errors are. The great thing about this, right, the structural variation, super useful for that. Same with Oxford Nanopore, a little bit different technology, but essentially the same idea. These are just going for longer reads. So I don't know what PacBio is up to, like what their longest reads are at this point. They have relatively low throughput again, just like PacBio does, but they sort of expanded. So we have a gridiron in the Genome Center that can get you 17 to 40 gigabytes. So it's lower. This is per flow cell, which is all I talked about with NovaSeq. So the gridiron and Prometheon are running multiple flow cells. Gridiron, it's a different kind of flow cell than Illumina's, but these will run multiple flow cells at a time. So that does increase your throughput, but the cost does not scale favorably for these compared to Illumina. So you can get those much throughput, but it's expensive. That said, Oxford Nanopore and PacBio have made big gains in the last 2 to 3 years, and so lots of the field is going in this direction. Part of a big genome project that doing almost all their sequencing on either Nanopore or PacBio with high-C as well. Okay. Okay, so now that's background on the technologies that are going in. So as a bioinformatician, what do you get? You get, like I said early on, you get the ACs, Gs, and Ts, and some indicator of the quality of the base pair calls. So the analysis tools, especially because the technology is continuously evolving, the analysis tools are trying to keep up. So there's not always a standard practice for how to deal with the data. Okay. The biggest differences are today are long read versus short read. A lot of the methods developed for short read because Illumina is so pervasive for so long, you know, they're not really useful. You have different methods and you have different problems associated with long reads. Okay. And then the wet lab methods that you use to generate the data can limit the available or appropriate analysis methods. So planning those experiments from the beginning is super important. For example, if you want to analyze allele frequency data, RNA-seq may not give that to you because that's going to tell you know, your gene expression level and your allele frequency can be conflated. Big time computational resources needed for these kinds of analyses. You're talking about having millions, potentially billions of reads that are hundreds of base pairs long, and you want to map those, find out where they belong in a genome that is millions or billions of base pairs long. So that is a lot of comparisons to make. The local resources, like on your laptop or in your lab desktop computer, are almost always insufficient. Not saying you can't do it. You can do it. My lab, Dr. Casto, Dr. Fujita's lab, we have dedicated machines in our labs to do this kind of stuff. Partly because the more computational resource you have, well, because our labs do de novo sequencing, new genomes, it pays to have that hardware. But for most people, it doesn't. You can just use the Exceed resources, and that's what we're going to do in our example. The downside is sometimes the tools aren't available. That is even an issue in the stuff that we're going to do today, because my favorite software for trimming reads is not on Bridges 2. But the other thing is that the runtime, there's a limit of 48 hours. So especially when you're doing genome assembly, which we're not going to do, but if that's a project, if you're working on a vertebrate-sized genome and you want to do assembly, you may need local resources because of the time limits on Exceed. So sequence quality control. What are the questions? You want to know if you got contamination in your sample, if there are adapters. Remember I talked about the indexing and primers and stuff. Are those in the reads? Because if they are, you're going to have trouble mapping. And then, especially for Illumina, is there a decline in the read quality at the 3' end? Okay. The creators of FastQC, which is the software that most people do to evaluate the quality of their reads, has a great website talking about next-gen sequencing quality control. So identifying QC, like I said, we're going to use QC issues in the raw reads. We're going to use FastQC. And then to fix those issues, there are several options. My favorite has been Trim Galore. Maybe my favorite, I've used it the most. But then Trim-O-Matic is also super popular, and that's the one we're going to use because that's the one that's available on Bridges 2. So what does Illumina sequence look like? Paired end sequencing comes in 2 matching FASTQ format files. They look something like this. I'm not going to walk through this whole thing, but it's every sequence read is represented by 4 lines. The top line is like the header in a FASTA file, gives information about where did this thing come from on the flow cell. The second line is the actual read sequence itself. The third line is almost always just a plus sign. Okay, it can include descriptors, but I don't think I've ever seen one where it actually that line was used. Okay, and then the bottom line is the quality score, and they line up with each of the base pairs in the sequence read. So the quality scores are based on ASCII encoded characters. So the equal sign, if you translate that into its ASCII code, corresponds with a number. So it's explained here, you know, how does that translate? Want to know more about, you know, sort of history and where these numbers come from, you can look at the Wikipedia page for FASTQ format. The main thing to know is that Illumina transitioned, it's been a while, maybe 10 years ago now, from version 1 to version 2, and you see the quality score notation changed. There are some telltale signs where some characters are used and others are not in one version versus the other. So most programs, like the one we're going to use, Trim-O-Matic today, it will recognize which scale the data is on automatically. Sometimes it guesses wrong, but so it's good to know. Any old data, like today we're working on old data, so it's a little ambiguous. You couldn't make an assumption that it was on the new scale just because the data we're using was collected a while ago. But any data that's if you collect new data, you can assume it's on the new scale. All right, so first thing that we're going to do, hopefully at the beginning you went ahead and downloaded your GVA data tarball, and you're going to have to move that to your home directory on Bridges 2. Okay, so use your Globus. There we go. So you should, on Bridges 2, move GVA data tar.gz over there. And then you're going to have to log in on your terminal. Let's see if I'm still logged in. Yes, I am. Okay. So this is logged into Bridges 2. And you can see I moved GVA data tar.gz over here, and then I did, whoops, well, right, you would do tar dash, we want to extract. It is gzipped. We want verbose to tell us what it's doing, and tell it the file name, GVA data dot. Okay, if you execute that, I'm not going to do it because I don't want to overwrite my folder here. It will create the GVA data folder for you, right? Because that's extracting that. And then if you go into that folder, I've already run some FastQC here, but what you're going to see is 3 files. And I think, yeah, I've got them on the PowerPoint. So your GVA data folder will just have these 3 files in it. So hopefully that goes smoothly. Next step, what's in the files, right? So remember how you look at those. If we look at the first and the second, oops, okay, let's. Okay, so that's the first one. That's the second one. So what I'm asking is look at the first 2. And you'll see that they are identical, meaning they came from the same spot on the flow cell. And this is read 1 and that's read 2. Okay. And then you could do the same thing for the last read using tail. And then how many reads do these things contain? Okay. So I'm gonna we'll work on this in class, and you can figure that one out on your own. Okay. Next step, right, our overall goal for today is figure out if these sequences are any good. So like I said, we're going to use FastQC. This is what pretty much everyone uses to analyze whether Illumina data are any good. I'll give you links for the online documentation for FastQC, and then some examples that they have there for what how to interpret good, bad, adapter contamination, things like that. But how do we implement it? So first thing that you'll want to do is start an interact session. And you don't have to do it for 90 minutes. I'm just said that's like, that'll take up at least the whole class period. But these jobs will finish much faster than that. So I'm just going to do interact for now. We'll see. Shouldn't take too long. Okay, so now we're on a compute node. And remember, to get access to software that's been compiled to work on bridges, we need to load the module. So you can do module load or ML fastqc. The capitalization is important there. So now we have access to fastqc, and then running it, super simple. Right, we just do fastqc on whatever our file that we want to run. Okay, so fastqc on that one. And then we will also run fastqc on the second one. Okay, so and then I'm going to quit my session for the moment. That way it just saves on our allocation. Question is, should we trim the data? Well, how do we actually look at the data? So this just gives you some background on what to look for. If you follow the link here to the online documentation, you'll get to the same explanations here. But the one that I look at first and am most interested in are the per base quality reports. How do we get there though? So look at what fastqc created, and then let's think about how we can view it. Whoops, let's do well. Okay, fastqc created these two files. Let me blow this up a little bit. Because remember, the only things we had at the beginning were the NC, this first FASTQ file, and then the second FASTQ file. So now we've got these HTML and zip files. Well, what you need to do is move those back onto your local computer so that we can view HTML. So if you use Globus to drag those back to your local computer, like I have done here. So this is my, I'm only looking at read one here. You should look at both. But when you double click on the HTML, it's actually accessing files in that zip folder. So you need both the zip file and the HTML. And here, like I was talking about earlier, is the standard profile, right? Nothing surprising here about an Illumina reads. It's got it kind of color coded, right? Good reads are in the green, or good quality scores. Back up a step. On the x-axis is the position in the read. Okay, so these are 36 base pair long reads. These are, like I said, these are older data back when Illumina, this was the maximum read length for Illumina, was 36. Okay. Nowadays, the x-axis would be 0 or 1 to 150, for example, but you would see the same profile basically, whereas you get out to 140 base pairs or so and things really start to trail off. in terms of their quality. Okay. The little icons here, most of these things, the default reading of quality by FastQC is saying we don't have too much to worry about. There's some issues with per base quality at the end. That's why it's giving us a caution here. The GC content, it's a little bit shifted off of 50/50. This is, again, this is a caution. It doesn't mean you can't use it. Sometimes the GC content being off is an indication of contamination, or you've got a bunch of adapter sequence in there. When the overrepresented sequence, though, it says we've got 53,000 that are all A, and that's the most common overrepresented one. Okay. So the main thing, we might want to get rid of those. The thing about overrepresented sequences, when you see something like this, is, well, you don't know where it came from, but this is not a case where I would be overly worried, because when we go to map, this is not going to map to the genome unless there is a sequence like that in the genome, which seems really unlikely. So how much work you go to eliminate a case like this depends a lot on the exact experiment and what you're trying to do. But what's more concerning is if you see overrepresented sequences that are like 4 or 5 base pairs long and could potentially be, say, an index primer if you use them, or an adapter sequence. Now we have pretty good, like this is saying it's not recognizing any of the Illumina adapters, which it has a database that it's checking standard Illumina adapters. And so it's saying it's not seeing overrepresentation or contamination by adapters, which is great. So there's not a whole lot to worry about here except for the ends down here that have really low quality. So go back here. Should we trim the data? Well, probably, at least for base quality, we should. And again, why? Well, when we try to go map a read, if it's got a whole bunch of errors down here, those are going to be mismatches to the reference genome that it's going to say, okay, the quality of this this read compared to this part of the genome is low, and so this must not be where it matches to or where it maps to. So we're going to decrease our mapping by having all these errors in the reads. Or potentially even worse, if it calls it, if it does map to that thing and it calls a variant that wasn't actually a variant, it was an error in the read. That's even worse. So we're going to try to improve the quality of those reads before we do the mapping. Okay. So this thing, nearly 1.5% of all the sequences are A's. Again, depending on what you want to do, is how much effort you want to go. Now, FastX toolkit, great toolkit, but it's not so good for dealing with paired end sequences because the trimmer there will trim read 1 and not retain the information with for which read it matches. So you can end up with a read 1 that has no read 2 partner, and that causes problems for downstream analysis where you're going to feed in paired end data. If the software doesn't find pairs, then most of the time it fails. Okay, so that's an issue with some of the FastX toolkit. Trim Galore was paired end aware, so it would keep the matches. But Trim Galore has been my favorite, but when they discontinued bridges and moved to bridges 2. I think because some of the components in Trim Galore weren't multi-threaded, so they didn't really speed up very well, or they didn't take full use of the infrastructure. They haven't ported that over to bridges yet, so we can't use Trim Galore. Okay, that was a bummer when I found that out. But it's okay. Trim Galore would have made it really easy to remove all those A's because it has an option that will let you specify exactly which kinds of overrepresented sequence to get rid of. But Trim-O-Matic is also really good, and it is available on Bridges 2. So we're going to use Trim-O-Matic to clean up our sequences. If you want to go to the manual here, I am going to give you the example command line for how to run Trim-O-Matic, but let me just show you. I don't think I still have it open. Do not. So this is the manual for it. These are all of the trimming steps. These are options for ways to trim. You go all the way to the bottom. Nope. So there are examples here for command lines. And you should always read the manual for software that you're going to deploy. Okay, for the moment, you can sort of take my word for how to run it and what it's doing. But it's always good if you're going to deploy this for your own research. You should definitely go read the background and the manual for how to deploy this. All right, so our goal for this part of the exercise is to trim the low quality reads. So we want to start an interact session. We're going to load the Trim-O-Matic module and then trim the low quality bases. And I'm giving you the command here. All right, so let's go over here. And I'm going to cd into, actually, I'm going to make a directory, mkdir. I'm going to call it trimming. The trimmed data there in my directory is just because I needed to make sure Trim-O-Matic worked on Bridges 2 earlier. So I'm going to make directory trimming. So now I'm going to cd into trimming. And now I'm just going to copy this thing. That way I'm sure I don't make any typos. Whoa, I didn't mean for it to execute. Must have copied the enter. But I copied the enter there, and so it already executed, and that was really fast. Oh, I see. It was really fast because it has an error. Ah, I see. It's because I went down one directory, so I'm in the trimming directory and my data is not in this directory. So I'm going to stick the path, right, because my data is up one directory. So I'm adding .. / before the file names for my input. I'll keep everything else the same, hit enter. And so this time it's working. Okay, so it says quality encoding detected as FRED3. That's what I was talking about. This is old. That's the older scoring scheme. So that's the quality scores it detected. And so here, well, there's your answer to the earlier question. This is one way. And lots of software will tell you what the input was before it gives you the output. But, earlier on, it was you could just use grep, sort of like we've done with FASTA headers to see how many sequences are in a file. You can do the same thing with FASTQ. But there were 3.8 million reads going in. how many times did both pairs survive our trimming? We are left with 2.5 million, so 66%. Now, sometimes it only kept the forward read, sometimes it only kept the reverse read. And then sometimes both of them were dropped, okay? So this should have generated. So now I have 4 trimmed reads files. So this has my potentially error corrected or filtered based on quality is really the right way to say it. So we've trimmed these reads based on quality score. And this says read 1 paired, and the P and the U are for paired and unpaired. So really the things that we have left for paired end reads are that file and that file. So in order to include these reads, we would have to map these as single end reads because they no longer have pairs to match to. All right, so just dissect what we just did a little bit based on the command we ran. So Trim-O-Matic said use that program. You know what, I don't think I did the ML Trim-O-Matic because I had done that before I started the recording. So you don't forget step 2 there. If you're getting failure like command not found or something, you have to do ML or module load Trim-O-Matic. The T has to be capitalized there. Okay, so let's call Trim-O-Matic. We're in paired end mode. Then we specify the 2 input files. That's where I originally got an error because you need to include the path to those input files. Then the base out option is saying what's the base name you want to give to the output files. And so that's why it says trimmed reads _1p and so on, because the base name was trimmed _reads. This Illumina clip says look for Illumina adapters and cut them off. Now our FastQC didn't say that they were a problem. It never hurts to keep that in there because it'll just eliminate it if it finds it. These things will cause reads to map just as bad or worse than poor quality. In fact, poor quality more often gives you variants that aren't real, whereas this just having adapter sequences in there just causes the reads to not map. This method of trimming, the trailing 28, says clip the bases from the 3' end. So the end of the read, starting there, if anything is below a quality of 28, chop it off. Okay, and then the minimum length is 29. So if you're chopping base pairs off the end and you get to 29 or less remaining, throw the whole read out. Okay, at that point it's too short. It's not going to map uniquely. We don't want to include that in our analysis anyway. All right. So last things, I'm going to turn you loose to do these on your own. You should run FastQ on these reads. So FastQC trimmed reads 1p. Okay, so that finish. Right, so now it generated my FastQC results for that. You should move those onto your local computer and look at them and see how did that go? Like, am I happy with my reads now? And I'm ready to move on with mapping. Okay. Whether or not, right, you would be happy with those reads, there's always a trade-off in how strict am I going to be with my read quality versus how many reads am I going to throw away. Just in this, the method that we used here, right, we used trailing with a quality of 28. We threw away what? 34% roughly of our reads, right? The one where both pairs are surviving is only 66% of what we started with. So we threw away a lot of data. Now, a bunch of that is in the single end stuff, but still, the question is, and then, you know, go look at the quality of what you're left with. Is that quality high enough, or should I throw away some more, or should I not correct? Maybe I shouldn't trim so much. I'm giving you another example here. This is another method. Instead of just trimming stuff off the end using the trailing method, Trim-O-Matic has something called a sliding window approach, where it calculates the average threshold. So now it's no longer if an individual base pair drops below 28 in the trailing, it cuts it off. Here we have a sliding window, and you can specify the size of the window, and it calculates the average score in that window. So maybe that would be a better way to cut stuff off the end, or at least know where to cut on the end, because rather than having an individual base pair drop, it's going to consider a whole window. And maybe correcting reads that way will give us cleaner reads. If it gives us cleaner reads, it's probably going to give us fewer reads. So again, that trade-off. All right, so we'll work on some of that stuff in class and talk more about how do you evaluate, you know, your trimming results. All right, that's it.